Dear students, good evening everybody. Welcome to NEET Bioforum channel. Today we are going to discuss about the chapter Molecular Basis of Inheritance or Molecular Genetics. So in this chapter we are going to discuss about the components of DNA, that is molecules of DNA. That's why this chapter is known as Molecular Basis of Inheritance. Okay, this chapter is related to DNA, RNA, proteins. Okay, then let us see one by one. So in this chapter the DNA, so the many processes going on in this chapter. That means DNA replication is there, transcription, translation, protein synthesis. So these all the processes are there. Then first let us see what are the types of replication. So there are three types of replication: dispensive replication, conservative replication, semi-conservative replication. So these are the three types of replication. So first of all we will see the history of DNA. So at first the DNA was identified by Friedrich and Mischer. So Friedrich Mischer, you can remember the food items. One is fried rice, another one is mixer. Okay, so you can remember these food items, means you can remember the names. Okay, Friedrich and Mischer. Okay, so the first they observed in the cell the nucleus is present. So here the nuclein is present. So the nuclein, it is the brain of the cell. The nucleus is the brain of the cell. They observed like that. Then the nucleus contains nucleic acid. So many years back in 1889, okay, so 1889 Altman observed this nucleic acid is named as DNA. Okay, so at first the DNA was identified by Friedrich and Mischer. Okay, then so many years back only they observed, who observed? Friedrich and Mischer observed the cell contains a nucleus. So it acts as a brain of the cell. So it contains nucleic acid. Okay, it contains nucleic acid. They named as nuclein. Okay, so then afterwards in 1889 Altman named this nucleic acid as DNA. Okay, so then next we will see the structure of DNA. So the DNA consists of, it has pentose sugar. So 5 carbon atoms. Then this pentose sugar may be DNA or RNA. Okay, so DNA means deoxyribose sugar, deoxyribose nucleic acid. Then RNA means ribose nucleic acid, ribose sugar. So deoxyribonucleic acid, that means in here D is present, isn't it? So devoid of oxygen. Okay, absence of oxygen. So devoid of oxygen. Here one oxygen is absent in the deoxyribonucleic acid. Then it has nitrogenous bases. So 4 nitrogen components are present. So here in DNA, adenine, thymine, guanine, cytosine is present. In RNA, instead of thymine, uracil is present. In RNA, instead of thymine, uracil is present. Okay, so the adenine, so it consists of 4 base pairs: adenine, thymine, guanine, cytosine. In DNA, in RNA, instead of thymine, uracil is present. Okay, then so here in DNA structure 5 carbon atoms are present. Then phosphate group is present. Then it consists of nitrogen bases. So in the structure of DNA, so basically the DNA is double stranded coiled structure. So it consists of, it is made up of 5 prime. It consists of 5 prime and 3 prime end. Okay, so one strand consists of 5 prime and 3 prime. Another one also 5 prime and 3 prime. Okay, so they are anti-parallel to each other. They are anti-parallel. Okay, so they are anti-parallel to each other. The adenine always pair with thymine. It consists of 2 hydrogen bonds. It is known as purine. guanine always pair with cytosine. So it consists of 3 hydrogen bonds. It is known as pyrimidine. So 2 hydrogen bonds. Purine consists of 2 hydrogen bonds. Pyrimidine consists of 3 hydrogen bonds. So in DNA, there is a special type of protein that is called a histone protein. Okay, so histone protein is present. The histone is positively charged. Histone is positively charged. Why it is positively charged? No, because of amino acid component, that is, it consists of 8 units of octamer. So octamer, anything which is related to octopus, for example octopus, 8. Okay, so it is 8 consists of octo means 8, isn't it? So 8 units of octamer. Then so it is the components of DNA. Then next we will see the RNA. Okay, so the RNA it acts as a catalyst. Okay, what is meant by catalyst? No, so it, for example, enzymes or substance. Okay, any substance or enzyme, it helps to speed up the reaction. Okay, so in the RNA, ribose enzyme is present. So it helps to speed up the reaction. Okay, so what makes this RNA much distinct from DNA? Okay, so because the RNA helps to bind with the DNA for the expression. Okay, then it takes place. It helps in the major role in the translation and protein synthesis. Okay, so in the translation and protein synthesis, in the next part we will see. Okay, then properties of DNA. What are the properties? No, it helps to replicate. It replicates itself. Okay, so replicates itself means one DNA is replicate into 2 DNA is replicate into 4. That is known as replication. Okay, then stability. The DNA has stability. So stability means, for example, right from young stage to till the end of the life. So our DNA has nitrogen bases. So that nitrogen bases never change. Always adenine pair with thymine, guanine pair with cytosine. So the base pairs never change. So it's stable. So the DNA is stable. Okay, so then information storage. So the DNA is good source for a information storage. So the DNA only store all the information. For example, her hair is like that. So she was smiled like this manner. Okay, so the DNA stored all the information. So the DNA act as a information storage. Okay, so these are the main properties of DNA. DNA replicates itself, stability, so it acts as a information storage. So in this today's topic, we discussed about the introductory session. In the next part, we will see the next topics. Okay, thank you.